Hello, junior cell biologists. Welcome to Module 2B, Chapter 3, the cells. This is cells 5. This is the last video in the series of cell videos. And in this video, we're going to talk about protein synthesis. So if you already know this, you know the central dogma of biology. You have DNA in the nucleus. The DNA gets transcribed into RNA. That's transcription. And then the RNA goes out of the nucleus through these nuclear pores and the RNA then gets translated into protein on the ribosomes, and the ribosomes are structures that live on the ER. Okay, this is a cartoon of the cell cycle. So we covered this in the last video, but in the cell cycle, wherever you see growth, you have to, when you grow, usually you're making new proteins, and that would include what we're talking about today in the video, which is protein synthesis. Here's our cartoon again. Here's the rough ER. Remember, the rough in rough ER means that the early scientists that were looking through a microscope and saw these dots on the ER but didn't know what they were, they just said they were rough. And then as science progressed, they knew that the rough was actually a ribosome. That's where you make proteins. So here's our cartoon for protein synthesis. Okay, remember this big purple ball here. This is the nucleus. And inside the nucleus, you guys know this, there's DNA. DNA is a double helix. It shows it as a double helix here. Now this is where you don't want to get confused between DNA synthesis and protein synthesis and DNA replication. So in the previous videos, we talked about DNA replication. So you're a mother cell and you need to make two new daughter cells. You need to duplicate, aka replicate your DNA so that each daughter cell gets a complete set of DNA in chromosome form. So actually the first step in DNA replication is that you unwind the DNA. And then in DNA replication, you make two new strands from your template strands, which were the original strands of nucleotide in DNA. So protein synthesis and DNA replication, they start off kind of the same way. You have to open up the double helix. But protein synthesis is different than DNA replication. So this cartoon is showing protein synthesis. So it shows the DNA has unwound. So you've got one strand of DNA here, another strand of DNA here. And then this strand of DNA is your template strand. That means that when you make your RNA from DNA, which is this blue ribbon, when you make your RNA from the DNA during transcription, that you use this strand of DNA to put your complementary nucleotide in the RNA. So for example, here's the sequence of nucleotides in DNA. You see here there's an A, there's a T, there's a C, etc. The complementary nucleotide to A in RNA is U, because RNA doesn't have a T. So usually in DNA, the A binds with the T, but when you go from DNA to RNA, the A complementary nucleotide to A in RNA is a U. So then you can see here's an A, RNA makes a U, here's an A in DNA, RNA makes a U, etc. And then so you build this RNA strand, and it's actually really complicated. So you build RNA first, and then you make messenger RNA, but we won't go into the details, the molecular details of that. But from DNA, you make RNA. And in this cartoon, they're calling it messenger RNA, because you're bringing the message to make a protein. Okay, so this, you can always tell RNA if you see a U in there. So this whole strand is messenger RNA. And then and that's the process of taking DNA and making RNA. It's called transcription. So DNA gets transcribed into RNA, then the RNA, they're just showing it as a blue ribbon here, goes out through a nuclear pore into the ER, finds this thing called a ribosome, and the RNA, which is this blue ribbon here, this is the RNA, gets translated into a protein. And here's your growing protein chain. So these are the amino acids that make up the growing protein chain. So alanine, phenylalanine, serine, glycine, and methionine. And then all these others are not named here in this cartoon. And it's quite a complicated process, so we'll go through it kind of one step at a time. So we talked about this messenger RNA here this messenger RNA will specify one polypeptide. So a polypeptide is just, it's the precursor to a full-grown protein. So polypeptide. It's made on the DNA template. We talked about this messenger RNA leaves the nucleus, goes to a ribosome, and starts translation. And then number 3, this incoming transfer RNA. So we have RNA, we have messenger RNA, we have transfer RNA, we also have ribosomal RNA. But in this cartoon, the transfer RNA with its anti-codon sequence finds the codon sequence on the messenger RNA, and that transfer RNA will bring in a single amino acid. And then that single amino acid, this amino acid coming in here will get hooked up to alanine, and then the transfer RNA will leave and go back to its transfer RNA home. And so this just shows the direction of the ribosome. So the ribosome is moving this way, and on each piece, 3 nucleotide piece of the messenger RNA, you have a codon. And that codon, like I said, will pair with an anticodon that's hooked onto transfer RNA. Transfer RNA carries an amino acid. And then you slowly build your polypeptide. Okay, so DNA, a lot of people like to say DNA is the blueprint for making proteins. So DNA in the nucleus here, this is the blueprint to actually make the messenger RNA, and then the messenger RNA makes the proteins. And then definition of a gene for this cartoon is a DNA segment that carries a blueprint for building one polypeptide. Proteins have a lot of different functions in the cell. They could be fibrous or a structural protein. They're like building materials for cells. Globular or functional proteins act as enzymes. And enzymes, as we've talked about before in previous videos, are biological catalysts. That means they make reactions happen quickly. Okay, so here's our cartoon. We'll go over it a little slower, one step at a time. We talked about this with the cartoon is enlarged, so you can see this sequence of DNA. This is the template sequence of DNA because you're going to make your messenger RNA from this strand of DNA, not this strand of DNA. So you'll make your messenger RNA, and there's a whole bunch of cool cell biology and biochemistry that we're not going to go into, but there's like a RNA polymerase that will make this RNA strand by putting a single nucleotide at a time onto the strand, and this single nucleotide will correspond with the nucleotide in DNA. Okay, and that's happening in the nucleus. And then out here in the cytoplasm, we see this thing called a synthetase enzyme. And this synthetase enzyme will take one of these guys, it kind of looks like a coat hanger sort of bent, one of these guys, a transfer RNA, and it will hook, the synthetase enzyme will hook a single amino acid on this transfer RNA. And then this transfer RNA has this anticodon sequence. It's not labeled here, but it could be like ATG or whatever it is here. but this anticodon will recognize a codon sequence on messenger RNA, and it will bring the amino acid into the developing polypeptide. So remember, a string of amino acid make a polypeptide, and then if you trick a polypeptide out, you can have a protein. Okay, so it says here the correct amino acid attaches to each species of transfer RNA. So each species of transfer RNA just means it has this, would have the exact same anticodon sequence. Okay, protein synthesis. Protein synthesis. DNA information is coded into triplets, and that's called a codon on the messenger RNA. So triplets, obviously triplets, 3, contain 3 bases, and they code for a particular amino acid. For example, we don't have it here, but if there was a DNA sequence of AAA, so if you could find a strand of DNA that had AAA, that would down the line code for the amino acid phenylalanine. So let me ask you this: if DNA has AAA, what will be the transcript from a DNA sequence of AAA? That is to say, DNA will get transcribed into what 3 nucleotides in RNA? If you said DNA sequence of AAA will give a UUU sequence in RNA, you'd be right. And then that UUU in RNA is the triplet code that will code for the amino acid phenylalanine. We've talked about this, ribosomes, most of them are located in the cytoplasm. DNA never leaves the nucleus in an interphase cell. So there's some fun cell biology factoids for your dinner party. DNA requires a decoder and a messenger to build proteins, and that function of being a decoder and messenger, that function is given to RNA. Okay, so protein synthesis involves 2 major phases, transcription and translation. Remember, I told you that transcription is a process where you transcribe DNA into RNA, and translation is the process where you translate RNA into a polypeptide and then a protein. So transcription, we've talked about that. It's information from DNA to the complementary base sequence of messenger RNA. So AAT, the complementary base sequence in RNA is UUA. So that's just kind of learning how to use the jargon. And we talked about this. If you are a triplet, so you can be a triplet here in DNA, and then when you make the corresponding triplet in RNA, that's called a codon. And this codon will specify a particular amino acid. So codons then, like I just said, are 3 base sequences on messenger RNA. So here's an example of transcription. So DNA triplets. So on DNA, if you had AAT, CGT, and TGC, when you made your messenger RNA codon, remember, A in DNA would bind with a T, but not in RNA. So you'd have an A has a complementary binding agreement with U, A with U, T with A, etc. So the codons are, you could also call them triplet codons if that helps you remember that it is a triplet sequence. And that's basically that's the genetic code of life. That's kind of cool. All right, so let's see. So we've kind of talked about these parts. I'm looking to see if there's anything that we haven't really covered. That looks good. Okay, protein synthesis. How is it different from DNA? So first of all, a couple of factoids. You need to know that RNA is single-stranded, DNA is double-stranded. RNA contains the sugar ribose. DNA contains the sugar deoxyribose. And we covered this when we talked about DNA when we did the chemistry section. So if you forgot about this, you can go back and review the chemistry section from these video lectures. And then we talked about this earlier. If you are RNA, you have uracil instead of thymine. So you have U instead of T. All right, and we talked about this guy's transfer RNA, this thing that looks like the bent coat hanger. And transfer RNA transfers appropriate amino acids to the ribosome to build the protein. So you've got your, well, let's look at this guy. You've got your amino acid here on the tip of this bent wire transfer RNA. This is the anti-codon sequence on transfer RNA. This is the codon sequence on messenger RNA. So this, and where did this messenger RNA come from? It came from 3 nucleotides on DNA. So the DNA was transcribed into this sequence on RNA. This is called a triplet codon. The transfer RNA, did I say this was transfer RNA? The messenger RNA. And then so the messenger RNA has the triplet codon. The transfer RNA, the bent hanger, has the anti-codon sequence that will recognize the codon sequence. And then this amino acid will be here, will begin synthesis of your polypeptide or protein. And then here's the second amino acid coming in, and it's got its anti-codon sequence. Here's the codon sequence on messenger RNA. And then when this guy docks, these two amino acids will be linked together through a dehydration synthesis. And we talked about that when we did the chemistry videos. And then over time, you'll build this really long strand of amino acids and you'll have a protein. So we've talked about messenger RNA, transfer RNA. Here's ribosomal RNA. Ribosomal RNA just helps form these ribosomes. So that, remember, this big orange thing is a ribosome. Okay. So this just enlarges the picture that we've talked about, enlarges it a little bit better. So remember this big orange thing? Here's the ribosome. It's made of protein and ribosomal RNA. This blue ribbon is the messenger RNA, and it's got, on the messenger RNA, it's got these triplet codons, and then the triplet anticodons on the transfer RNA will complement each other, form an association, and you'll bring in one amino acid at a time. So let's see what it says. Number 3, incoming transfer RNA, it's the bent hanger, recognizes a complementary messenger RNA codon. So complementary, that word just means that the two bases they complement each other. So remember, in DNA, A always binds with T, G always binds with C. In the RNA world, G always finds a complementary partner with C, and then U finds a complementary partner with A. So that's where the word complementary comes in. Okay, and then they're giving you the specific names for the parts of this bent hanger transfer RNA. This is the transfer RNA head that has the anticodon that we've been talking about. And then this shows the direction of the ribosome movement. So the ribosome's moving this way, and then so that means that the portion of messenger RNA is moving this way relatively. So the ribosome moves relative to messenger RNA. Okay, so then number 4 says, as the ribosome moves along the messenger RNA, so the ribosome is moving this way along the messenger RNA while the messenger RNA is moving in the opposite direction, a new amino acid is added to the growing protein chain. That's what this is. So when this transfer RNA docks here in this codon site, then these two amino acids will be linked via protein via a dehydration synthesis. And then step 5, the released transfer RNA. So after it delivered its amino acid. So now the transfer RNA releases itself from the ribosome, and it goes back into the cytoplasmic pool of transfer RNA. It gets ready to be recharged with that synthetase enzyme. Okay, so let's look at what we have with our little squares here. The protein synthesis, translation. So remember, this process that we've been talking about is translation. So you're translating the messenger RNA into this growing amino acid chain called a polypeptide. So amino acids are the building blocks of protein. So each one of these is an amino acid. So step 1, transcription. We talked about transcription. So in protein synthesis, step 1 is transcription. Step 2, the messenger RNA leaves the nucleus and goes to a ribosome. We talked about that. Step 3, the incoming transfer RNA, that's this guy, recognizes a complementary messenger RNA, triplet codon, calling for its amino acid. Transfer A uses an anticodon, and it's carrying the amino acid. As the ribosome moves along the messenger RNA, so ribosome is moving this way along the messenger RNA, a new amino acid is added to the growing chain. And then when the transfer RNA has hooked its amino acid up to the growing chain, transfer RNA minus its amino acid goes back into the cytoplasmic pool